Right. Welcome, everyone, to this week's colloquium. The speaker is going to be Professor Vijay Balasubramanian, and the title of his talk is going to be How Brain Maps Space. Vijay is the Catherine and Mark Lasry Professor of Physics at University of Pennsylvania. If you go to his official Penn website and look at the thumbnail sketch of what he does for a living, he says he's all about how natural systems manipulate and process information and lead to new forms of organization. In fact, he does this under three subheadings. One is fundamental physics, and/or the nature of space-time and black holes and quantum mechanics thereof. Under the heading of biophysics, how the brain works in sort of almost every mode of operation that it is available to it: visual, auditory, olfactory, and more abstractly in statistical inference and machine learning. Namely, how do you go about in a so to say, principled way to infer quantitative models of real-world data? And you know, if you read a subset of the titles of papers that go under these headings, you get a sense that this is a guy he's bucking for the Renaissance man medal of modern science. He has done an absolutely amazing range of things in a spectacular span of intellectual space. I'll give you a brief CV. He was an undergraduate at MIT. Majoring in physics was not enough. At the same time, he majored in computer science and got a master's in computer science along with his physics degree. He went to Princeton, where he got his PhD. Says that I was his supervisor, but as John Schwartz said at some event about one of his students, I immediately thought the best thing for me to do was to get out of the way, and it's certainly what I did. After he graduated, he was a junior fellow at Harvard in the Society of Fellows, and he then moved on to the University of Pennsylvania as an assistant professor and rapidly rose through the ranks. And at this moment, as I said, he's an endowed professor of physics. In addition, at Penn, he's a member of the neuroscience department. He's the director of the Computational Neuroscience Institute. He's a stalwart of the Aspen Center for Physics. He's one of the general members. I'm sure he's been head of various subcommittees. He's an external faculty member of the Santa Fe Institute. Awards, well, he's a fellow of the American Physical Society. He was a recipient of the first prize for a paper on gravity of the Gravitational Research Foundation. And one thing that I like particularly is that he won University of Pennsylvania's award for distinguished teaching. And if you look at his, so to say, teaching record, you're impressed. What are the courses that he teaches to great acclaim? Theoretical and computational neuroscience, general relativity and gravitation, freshman seminars with titles like Big Bang and Beyond, The Order of Things, et cetera, et cetera, et cetera. Is a man for all seasons and all reasons. I have just one, I'll turn it over to him almost immediately. But as you can see, I have a great, I'm greatly impressed by and very respectful of the scope of his intellect. And I vividly remember when he came to Princeton as a graduate student, you know, on September something or other, there was the reception for incoming graduate students. And somehow we came together, started chatting, and it finished maybe 2 hours later. You know, it started with their string theory, then it became Wither biological modeling. He explained to me his ideas about Occam's razor, and eventually we passed on to literature, and we discussed a suitable boy at great length. And the only kind of paper that we talked about in those days, or the only aspect of intellectual life that has not been covered in his official publication list, as far as I know, is literature. But maybe he'll get to that at some point. Okay, so please welcome Vijay Balasubramanian.
Lovely introduction. I would say Kurt was a really wonderful advisor because, you know, You gave me lots of space to do whatever I felt like doing, and I really appreciated that. Can you hear me? Great. So I'm actually one of the official organizers here of the Adaptive Immunity Workshop. So I think original plan was to give a talk on statistical physics and adaptive immunity. And then I realized about half of you are immune people and be bored out of your skulls. Instead, I thought I'd give a talk on something else, hopefully new to most of you. The topic of my talk today is this: how the brain maps space. The details of what I want to mean by that and what I'll discuss will become apparent in a few minutes. So if we're going to talk about how the brain maps space, first thing is to discuss is what is space. As physicists, we all have a commitment to this. But actually, space is a more complicated concept. And since ancient times, we've kind of understood space as a sort of stage on which physical things are located. And if you go back, people have had many concepts of what space may be. Some have thought of space as infinite, others as finite, flat, round, continuous, discrete, centered on Earth, centered on sun, centered nowhere. And all of these kinds of ideas that we beat about in physics have been thought about elsewhere before. Space can also be abstract. It can be the cosmos as a whole, or a space of relationships between entities. For example, there is a diagram of the cosmos drawn by somebody about 1,000 years ago in Nepal, and it describes a certain kind of arrangement in some cosmic space of different kinds of entities that they believed then underpin the nature of the universe. You can also move between space. That's one of the characteristic features of space. And that too has been conceived of in different ways. In Mesoamerica and Australia, traditionally people thought of 4 cardinal directions: north, south, east, west. At various points, different places in India, there have been considerations of 10 cardinal directions: north, south, east, west, in between, up, down. You can think about this in different kinds of ways. Now, as physicists, our modern way of thinking about space has a lot to do, is due a lot to Descartes. And he gave us our modern notion of space as a stage for action, described with some coordinate system, with some number of dimensions, things of this kind. So we're really wedded to this way of thinking. It's true that it's been modified a little bit. Einstein, of course, taught us this grid kind of warp and bend depending upon this matter and energy. Even more recently, in the last 25 years or so, we understand that space itself can be kind of emergent, in the sense that it can be kind of a convenient way or effective way of describing some other complicated many-body dynamical system that doesn't have the space. Nevertheless, in all these descriptions, we tend to use this kind of Cartesian picture. There's a fixed thing, with some set of coordinates and directions and things of this kind. Now, if you're an animal, space is central to you. That's because animal life is practically defined by motion in space. If we didn't move, we would have to adopt the lifestyle of plants and use chemistry as opposed to motion to do the things we need to do to live. To move around and be an animal, there are certain things you have to be able to do. The first is you have to be able to learn geometry of new spaces. Having learned that geometry, you have to be able to imagine where you are in that space. And then you have to be able to imagine plans, make plans getting from one place to another place. These 3 functions are just absolutely critical to the existence of animals, all kinds of animals. The confusion or difficult part is inside your head, there isn't a map or a Google map or anything like that sitting there. Inside your head, here, this location, this is my living room, for example, is a kind of abstract pattern of neurons firing. Right? So there's some pattern. And these patterns are said by the professionals maintain your cognitive map of the world. And it is the map of location in both physical and abstract spaces that you can navigate in, spaces of shapes and things. If you navigate them in your head using this abstract pattern of neural firing. So in this sense, you could say that within our heads, space is always emergent. It's a kind of abstraction describing the interrelationship in sparks of activity between different neurons. So there's a whole large chunk of the field of neuroscience, many experimentalists and many theorists in it, which is devoted to the question of how does this happen that is so important to the existence of animal life. That's going to be pretty much the topic of the talk today. I'll only be able to give you a little glimpse of all the work going on here. I think a little glimpse with some detail possibly better than trying to give you a very tiny amount of everything. Now, I think essentially none of you are neuroscientists. So I think it's a good idea for me to start by setting some vocabulary, discussing some basics of the circuits in the brain that are supposed to produce this kind of map inside your head. So start with that. So this is your introduction to neuroscience as a field for this purpose. So the brain, like any other part of the body, is made of cells. And cells, like all cells, there's a membrane that surrounds some stuff inside, and we're eukaryotes. So there's a nucleus, and there's various kinds of chemistry and biochemistry going on inside. Neurons, in particular, are charged with computation and communication, and therefore they have various specializations that make it possible for them to communicate and compute. First of all, morphologically, namely in terms of their shape, there's a cell body. Coming out of the cell body, there are these things called dendrites. Dendrites are going to be the input wires of the neuron, where signals come in from elsewhere, from the world, or from other neurons. The neuron, the cell body, can produce signals. We'll discuss those in a minute. And they're transmitted down a long wire called the axon. And the axon then can produce some other terminals, some dendrites. These things have synapses on them. These are the famous synapses everybody talks about. The synapses communicate with receptors on the next neuron, and the synapses will release neurotransmitter to the next neuron. It's a chemical communication, and then take the signal down the wire. So that's how circuits are built. Now, neurons themselves are actually electrical circuit elements, like transistors, diodes, what have you. And this is how they work. They have a resting potential of minus 70 millivolts or so relative to the background. This is maintained by a set of active pumps in the cell body. As the ions leak, sodium, potassium pumps and things like that keep sort of equalizing the concentrations of sodium, potassium, calcium chloride, other kinds of ions in such a manner that they actively maintain this minus 70 millivolts. When there's input to the neuron, ion channels open. That causes more ions to flow. Basically, current flows in and out of the neuron, and the potential increases. If the potential increases to some critical threshold, a feedback process kicks in. That feedback process causes the voltage to pulse very rapidly and very sharply up to, let's say, 150 millivolts, at which point some other negative feedback kicks in, and boom, it crashes down to back to minus 70 like no time. So this whole pulse is like a millisecond long. And if you record electrically from a neuron, you'll see things like this. It'll bounce around, and boom, boom, boom. There'll be some pulses like these. These things are called spikes or action potentials. And essentially, everything you think is based on digital signals sent between neurons in this way. Right? If you threshold here, this is like ones and zeros. That's how you're computing. You're a digital computer. Now, back in the day, when they first discovered, this was first discovered, the idea was discovered by the Galvanis, the husband and wife team, who were, I think, in Bologna, and I think Volta or somebody just invented the battery, and they were cooking frogs' legs at the dinner table, and happened to some charge through a frog's leg, and it went kick. And they were like, wow. And that inaugurated the idea of animal electricity. There was electrical effects in animals. And indeed, this also led further to the idea that somehow life itself was based on electricity. With the idea of vitalism, that electricity was sort of the spark of life. So there were lots of crazy experiments, like they would, if you hung a criminal, this was done in Scotland, then pump the criminal, the dead body of the criminal, full of electricity, and the body would do this kind of thing. They would say, oh, wow, look, it came back alive. But of course, it didn't. What happened was that all the muscles, which are innervated by neurons, were moving because the neurons were getting excited. The electricity pumped into the body, the whole thing would sort of move. This is the source, ultimately, in literature of things like Mary Shelley's Frankenstein and the inauguration of a whole bunch of thinking, artificial life, and eventually leading now to artificial intelligence. Now, of course, a single neuron can't do much. Can't write poetry, can't write talks, like this. It has to be most of what we care about in animals is a collective effect of many, many neurons. Now, human brain contains about 100 billion neurons. So if it was really like it is like artificial neural networks, this gigantic mishmash of neurons connecting everywhere all over the place, we would have basically no hope of understanding this, right? Because it's just too complicated. But fortunately, in fact, the functions of the brain are specialized and localized to different areas. It's like when you write a computer program, you're well advised not to write spaghetti code. Right? You're supposed to write procedures that do one thing or another thing or a third thing, then hook them together through well-defined interfaces. The whole thing is under control, can cooperate nicely to do things. At the scale of the whole brain, that is how things are organized. For example, back here is the visual cortex. This is why, if you fall on the back of your head, you'll see stars. You have a bruise in your visual cortex, and the circuits aren't working that well. Over here is Broca's area. Broca's area allows you to produce speech, grammatical speech. This area is very famous because the modern theory of the localization of function in the brain is due to a certain Dr. Broca, French doctor, in the mid-19th century, who had a patient who had, I think, a spike or something go into this piece of the brain, and the patient was perfectly fine, except he couldn't produce speech. That led Broca to propose the modern idea of localization of function of this kind. So as you can see here, different kind of functions that you do are localized in different places. So, for example, motor control of the body, is localized in a strip that runs down this way. If you look even closer at those regions, you find that within those regions, it's not like there's a gigantic neural network in the modern computer sense controlling your whole body. Rather, circuits at the top control your knee, circuits here control your wrist and hand. If you're a human, there's a lot of real estate spent in the brain on your face and on your hands, because that's really important to us as primates. The hands, and as social animals, facial expressions are really important. This is also why, if you have a stroke, for example, over here, then the left side of your face will sag. The right side is perfectly fine. It's because of this kind of localization function. In fact, the localized damage the brain can produce these effects has been known a long time. So actually, the idea of localization of function in the brain goes back to the Egyptians. So this is something called the Smith Papyrus, written in 3000 BC, bought by Mr. Smith in 1860 or something. And this is a treatise, an ancient Egyptian medical treatise, in which the author is describing traumatic brain injuries and what to do about them and what their symptoms are going to be. So in this, there's the first glimpse of the human discovery of the localization of function. So today's talk, we're going to be especially interested in the area of the brain called the hippocampal formation. So deep inside here in the midbrain, there is this region called the hippocampus. And the hippocampus contains, and the regions around it, contain many of the circuits that facilitate things like memory, long-term memory, navigation, planning, and the mapping of the world. Today, much of what I'm going to talk about is localized in this region. Before I continue, I have to add one more thing. I described these different brain areas, and then I described how this brain area, which is that takes care of motor control, is further subdivided into many different kind of little pieces that do different control, different bits of your body. In fact, were you to look at any one of those areas, you would find a further specialization. This kind of specialization goes all the way down the cell circuit level. Here's the retina, probably the best studied part of the central brain. This is a beautiful picture drawn by Ramon y Cajal, essentially the founder of the field, in 1917. And at his level of precision, the retina is your classic three-layer neural network. The front end, there are the receptors, which transcribe light from the world into electrical signals. For the many of you who work on artificial neural networks, that's like a convolutional layer, bringing in some signal from the world. And there's a second layer of so-called bipolar cells. The bipolar cells do some sort of analog processing of signal. And then the third layer are the so-called ganglion cells. The ganglion cells, first of all, they're digital devices. They respond with spikes that go down the axons, which make the optic nerve bundle. But the important thing for us is there are many types of each of these cells. It's a three-layer circuit. There are three types of rods and cones, three types of cones and one type of rod. It's about 10 types of bipolar cells, 15 kinds of ganglion cells. And in between these, there are sort of lateral neurons that go sideways in the circuit. And there are 30, 35 types of these things. They're all distinctive, look different. And in biology, there's a rule, form follows function. So they look different, they're probably doing different. And indeed, if you measure their responses, they're doing different. And the biochemistry is different. But there's this repertoire. And this repertoire is largely conserved across the vertebrates, suggesting that there's something good about the repertoire in terms of facilitating vision. So an example, a question many physicists turned neuroscientists have asked, why do you have this repertoire, right? Why did you need more than one kind of cell, first of all? Because you could have built all of these out of NAND gates. If you do commit to having more than one kind of cell, why this repertoire, not some other repertoire of different kinds of feature detectors? I should say, these ganglion cells here are, for those who have done any computer vision at all, are like what are called feature detectors. They will detect like, edges and corners and left to right motion and little patches of color, things of this kind. So they basically take apart the world into all of these little kinds of visual features, send it back to the cortex, and the cortex reassembles everything you see about the world. Now, our interest today, given that prelude, is in the circuits that underlie our sense of space. Now, here is a very nice circuit diagram. So this is what we're going to be talking about, parts of this. This is a circuit diagram drawn by Jim Knierim, a professor at Johns Hopkins a few years ago. Let me talk a little bit about the different components of it. In order to make a map of space, you need inputs from somewhere. The one class of inputs you could have coming in through an area called the perirhinal cortex are sort of sensory inputs, things you see, vision, audition, things like that, associated with items, things in the world. It's a second pathway that has to do with scenes, where everything is laid out. Then there's another set of pathways, sensory input pathways, that have to do with motion, right? You're sending signals to your own body, telling your body how to move. So you already have internal information about your intent and how you should move. You can also feel your own body. So even in the dark, you kind of have a sense of how far you moved. So all of that data comes in a second pathway over here. These two pathways then go to the lateral side, the medial, the central part of an area of the brain called the entorhinal cortex. That's what we'll mostly focus on today. And this area of the brain is now known to play a very, very important role in producing both spatial maps and having maps of where things are in that world. We will focus just on the spatial maps. The data from that goes to what's called the hippocampus. The hippocampus has further sort of regions in it. And all told, the hippocampus does things like, being able to load maps in, remember things, remember maps, help you to make navigational plans, associate the plan you want to make with the other contexts that are relevant for your behavior, right? I mean, you're in some space. There's one context you may want to go that way. There's another context you may want to find your way that way. All of that has to come in somewhere, and that data, the context for your behavior, seems to come into the hippocampus and then affect how you make spatial map. That's all the areas of the brain that are involved. Then, as I said a moment ago, there are many types of cells involved. If I go look at just the entorhinal cortex, well, these areas, you will find, for example, border cells. We will mention more later. Border cells respond when the animal is in a particular part of the border of an environment. It's sort of a map of the edges of space. There are boundary vector cells that respond when your head is pointing at some angle towards the border. Then there's direction cells that just respond in a way that's related to which way your head is pointing relative to an absolute landmark. Speed cells, place cells, landmark cells, egocentric cells. That means responding relative to self, get to the right or left of me, and allocentric cells, responding if it's north or south, that kind of thing. All of these different kinds of things that you could imagine part of the repertoire of a computer or a robot you build to navigate space. There in the brain, people have carefully been mapping out all these different types of components of the circuit. Our goal is to try to think about a theory. I'm a theorist. I'm interested in a theory of the organization of these circuits. So overall, I work in many parts of the brain. I have a tendency to view the circuit repertoire of the brain as a kind of memory of the computations that have predictive value for behavior that you've learned over evolutionary time, and then encoded in the genome and the developmental program. That's the attitude I like to take. And then I like to ask the question, what organizational principles or laws control these computational and information processing repertoire? So that's the sort of broad goal of my group. Okay. A nice book for those of you who want some bedside reading. Peter Sterling, my mentor into neuroscience, Simon Laughlin, called Principles of Neural Design. It's very readable. It's intended for the general audience. You might enjoy that. That lays out some principles. So I'm going to talk about two of these principles because we won't have time to incorporate many more of these. One is the cost of computation. Here's your brain, and here's your laptop. Your brain is only 2% of your body weight. But it's 20% of your metabolic load. It costs about 10 times as much as muscle to operate your brain. So it's really expensive for you. On the other hand, compared to your laptop, it's really cheap. The brain is estimated to consume about 20 watts of power. This is like negligible compared to most of our devices. Particularly the laptops, our typical laptops these days consume 80 watts of power. They can't, well, maybe they can now write poetry. You know, that was because Google bought a city's worth of electricity to sort of do their processing in the back. You're not going to be able to do that very easily on your laptop. So there's a question of how you manage to achieve this kind of efficiency. A popular, very powerful idea in neuroscience has turned out to be that you achieve these sorts of efficiencies by basically adapting your circuits to the structure of data in the world, because that's where the stuff is. So roughly speaking, you know, in qualitative terms, you know, you can write this quantitatively in terms of information theory. The qualitative idea is something like this. The idea is, you know, if you talk about societies back in the day, we're all hunter-gatherers, and, you know, we did everything, dug the ground, hunted the animals, you know, the house and stuff like that. Then, and, you know, society progresses along, and economists, sociologists will tell you that eventually we specialize functions. You know, baker breaks the bread, shoemaker makes the shoes, what have you. Each unit in the society squeezes out more efficiencies, and the whole system works better. So people have applied quantitative versions of the idea, the organization of these kinds of circuits. So one very popular idea is a theory of the organization of neural circuits is they should be efficient with respect to the correct resource when they do the tasks that they perform. Here's that idea. The idea is that brains exploit the average structure of the world, efficiently allocate limited computational resources, maximize gain for the organism. You can see where you might have here beginnings of a theoretical formalism. You have a certain amount of resource, certain number of cells, certain number of ATP, the energy currency of the brain, certain number of amount of time, you know, whatever that may be, and you need the circuit to do its job quickly, effectively, and with minimal resources. And there are extensive bodies of work in computer science, statistical physics, information theory, control theory on trying to do that kind of thing with artificial systems. You can borrow from those frameworks to think about this problem. So very roughly, the way these ideas work is that you have some function you want to perform. Here's the space of vision, or some cognitive functions you want to perform, and you imagine, you select different architectural elements to perform little pieces of this task. That's like all the little units in society, all communicate in some way. And you ask yourself questions like, how should you organize the mosaic of things that perform functions in order to achieve the overall thing that you need to do? The second principle that's often very important is learning and self-organization. This is my daughter who's over there, second person back there. She was much smaller. She was always thinking. So yeah, here. And the idea in this is that you don't have to get your neural circuits to be perfect the time you've sort of wired them up at birth. But you can self-organize because neurons have synapses, can change in strength, reorganize themselves. So the idea then, the neural circuits self-organize their architecture through dynamics and through learning, and we'll do a little bit of both today, in order to adapt different kinds of variations in the world around the average that they learned over evolutionary time. Okay? So this is the broad body of thinking in how you think about these kinds of organizations of circuits. I'll take questions if you have any, and then I'd like to get into the actual problem of applying these ideas to the mapping of space by the brain. Wait, let's start. Yeah.
On the intro. So you said that in our brains, you have all this sort of different types of cells. Yeah. Are there brains of other animals where the organization is different? Where you
no.
Have this special, so you always...
This is a universal observation. C. elegans with 360 or whatever. How many cells does C. elegans have? 307 cells or something like this. They're all different from each other.
each has a function.
Yeah, each has a function. Right? That's not 100% true. Like the things that control, motion, there are similar neurons that control motions at different parts of the C. elegans, but they're basically all important. That's so for us, you can lose a significant fraction of your head and our brain and still function perfectly fine because there's much more redundancy for us. Okay, let's talk about mapping space. So once again, our problem is we want to talk about the sense of place, but where you are. And the question we have is what is place and how do you know where you are? Remember, I said that inside your head, here, any location, is some abstract pattern of neurons firing. Now, the approved way, working on problems in biology, physicists, is to first go look at the data, understand it, and do a phenomenology bringing up. But I'm trained as a string theorist, I have a tendency to do the reverse. So I'm going to do the reverse, and you'll see some good things will happen. So let's not look at the data yet. Instead, let's step away and ask, suppose you wanted to design a neural system to represent space. What should you do? It's useful sometimes to have that, partly because when you get data, you never get data. Experimentalists always give you data that's been processed through multiple layers of theory and understanding and expectation of what the world is like. You never actually look at raw data. You can get misled by the actual pre-processing of the data before it reaches your hands. Sometimes it's useful to step back, think about how should it be, and then go back and look at the data. That's what I'm going to do. So suppose your world was an 8-meter linear track, and for some behavioral reason, you needed to have 1-meter resolution. How could you design a system to represent this? One way you could do this is you could divide the world into 8 regions and then have so-called place neurons to fire when you are in one of these locations. So if we're talking about neuron 3, and the red dot is the mouse, you might hear brrr, and then it shuts up. So that would be an encoding, mapping of space. Depending upon which neuron fires, you know where you are. In fact, there are cells in the hippocampus that behave a bit like this. But we now know that those cells respond to lots of other things. They respond to context for behavior, they respond to smells and odor, what have you. It's more like they're very general context cells, and place happens to be one of the contexts that's relevant for your behavior. So we'll talk more about that later. If I'm a theorist and I say, well, there are more efficient way of representing space, because after all, we said that efficiency is somewhat important, well, we might try this. You might divide the room at the biggest scale into 2 parts, the left and the right, called the 0 and the 1 part, and subdivide by 2s, and here the 0 is in the left half of the room and the left half of the right half of the room. The 1 cell represents the right half of the left half of the room, the right half of the right half of the room. This is going to get confusing, but you get my point. You subdivide by 2s. At each scale, there are 2 cells. They respond sort of in a periodic, latticized way. That's kind of weird. This is, after all, a binary representation of space. This location is 0, 1, 0. This location is 1, 0, 1. This requires only 6 neurons, 2 less than what we had before. It's a bit more efficient. But actually, if the resolution was much finer, the room was much bigger, you would get an exponential improvement in the representational capacity of this code. And of course, you could use other bases like base 10, whatever you want to use. Okay, so that's the one-dimensional world. Most terrestrial navigation is in 2 dimensions. Let's think again what you would do in 2 dimensions. Imagine the world is an 8-meter by 8-meter square, that you need 1 meter by 1 meter resolution. If I have a place code, a unary code, number code, for the world, you'd need 64 neurons. Now let's try the analog of binary numbers in 2 dimensions. What you do is you divide the world into the room into 4 corners, green, yellow, orange, and blue. Then you subdivide recursively again and again by factors of 2. So for example, this yellow cell would fire if the animal were here, or here, or here, or here, that kind of thing. Right? And now, for example, the location marked by X is the blue, yellow, blue location on this map. So now, by combining the information from these kind of strange, periodically responding neurons at different scales, you could get a map of space with just 12 neurons, much, much better. And of course, your gain is huge if the room were bigger and your resolution were finer. This will be for a mouse or something. And you can use other bases like base 10. Next question is does this have any relationship to reality? So I have here a picture of recordings done about almost 20 years ago now by lab of Edvard May-Britt Moser in Trondheim, Norway, in the entorhinal cortex. So they poked an electrode into this area. And let's look at this picture. This is a picture of locations in the room where a mouse was standing and this neuron fired. Okay, so hotspots are telling you the number of spikes per second this neuron emitted as the animal wandered around this room. Notice the triangular lattice. You poke further in, and this is a paper that came out in 2012, Stencil et al., also the same lab. You find another grid. This grid has a finer scale. And then you go further, you find a finer scale still. You find a series of scales. Let me summarize the findings. First, grid cells in the entorhinal cortex respond when the animal is physically located on locations lying on a triangular lattice. Different neighboring cells have randomly varying offsets. So the neighboring cells are responding, with random phases relative to each other. As you go into the entorhinal cortex, grids increase in size. But the increase in size in an interesting way is a series of discrete modules in different periods. Okay, so I'd like to put to you that that looks a lot like this cartoon I was drawing. I didn't particularly try to optimize this cartoon. I said, it seems like it'd be more efficient to have a sort of grid-like representation, basically a base and number system. I would say that there is some sense in which the experimental data looks like some kind of two-dimensional fuzzy neural number system. Okay. How can we get further evidence for this idea? Earlier, I said that a popular paradigm the neuroscientists use is that the circuit should be adapted to do their job efficiently with respect to some resource. So we could ask ourselves, which number system, binary, decimal, etc., should the brain pick? What would be the most efficient thing according to whatever resources you think need to be conserved? I need to add a little point of caution here. Normally, when we think of base and number systems, we think integers, so base 2, base 3, base 4, etc. But that's just because we need to write the stupid thing down. In this situation, the base of the number system is simply the ratio of the periodicity at the different scales. And in fact, you could have different periodicities between here and here and here and here. You could have a continuously varying base. The base doesn't have to be integer. It can be irrational. It can be whatever you want it to be. Whatever it takes to do the job well, since you don't need to write the numbers down. Great. There's also constraints. When you do a problem like this, we're going to optimize this grid, represent space. But without constraints, you'll always get ridiculous answers. You got to ask yourself, what are reasonable constraints that are pertinent here? Here's one, very important one. Imagine first that you have this kind of grid organization in one dimension. The red cell and the blue cell responding to the left half, the right half of the room. And then, you have the red cell here, and then the red, finer and finer scales as you go down. The animal is here. I know red, red, red responded. Now imagine you get greedy and try to squeeze more resolution of the system faster by squeezing this grid in. Now consider this organization. In this organization, you have the red cell respond, and then the red cell respond. You don't know where the animal is. They could be here or there. In other words, the grid field, the ambiguities will arise in your code if, for space, the grid field width is too large compared to the next scale. So that gives you a constraint that the field width at one scale should be somehow smaller than the period at the previous scale. We'll come back to that in a minute. So you might be by now thinking, people who actually are biologists in the audience will be thinking this is all very cartoony, and start getting more serious. And yes, you do have to start getting more serious. So for example, real neurons don't fire in this nice square blocks. They'll have what's called a firing field or receptive field. You can model it roughly as a Gaussian. They'll mostly fire here, but there are little tails floating around. Things of this kind. But you can quickly convince yourself that a similar constraint applies in that case too. It's really a matter if the principal lobes of these different firing regions overlap or don't overlap. Okay, so let's keep this constraint in mind. And now I'm a theorist. What I'm going to do is I'm going to formalize the problem. Here's the entorhinal cortex, the medial entorhinal cortex. At this end, at the ventral end, we have wide grids. At the fine end, at the dorsal end, you've got fine grids. I'm going to parameterize these grids in terms of the width of the firing field, that's Li, at scale I or module I. And then the period of this is going to be lambda I. If I'm going to talk about grids in more than one dimension, strictly speaking, there are lattice vectors to worry about too. For the purpose of this colloquium, I'm sort of leaving those out. I'll mention them a little bit later. Okay. What are the relevant parameters we should extract from this that we should think about? So one parameter is the ratio of grid scales, lambda I over lambda I plus 1, those periods. Why? That's what I want to interpret as the base of this number system. Another important parameter for an animal is the resolution you have in space. Right? How finely do you describe space? Roughly speaking, that number is given by taking the biggest period divided by the smallest period, because that tells you how many pieces you can chop your arena into. If you agree that your biggest period is, like the whole space, right? That scale, right? It depends upon your home range of the animal, and then you have the fine scale, which you need to know your behavior. That's probably your body size or something, right? That ratio is the resolution. That's a fixed number that's given to you by behavior of the animal. That has to be fixed, and that, if you notice, is a product of these ratios of scales, telescoping product of this kind. Another parameter of importance for us is we need to be able to resolve space. Does that mean, well, take the whole region, lambda I, and then I chop it up into little regions of size Li. How many of these regions do I need? Well, I need enough cells, lambda I over Li, the number of cells I need. Now, strictly speaking, if I was doing this a bit more carefully, I'd have to remember the neurons have some noise, they have some overlap, etc. The reason I put a proportionality sign is the number, there's a constant here associated that you could associate sort of the noisiness in response to these cells, and I'm leaving that out for the purposes of this colloquium. And we're going to agree the periods descending in size, lambda 1 bigger than lambda 2, etc. I've written all these formulae for one-dimensional grids. If you wish to write them for a d-dimensional grid, you raise these things to the power d, because instead of lengths, you have to talk about areas. And there may be some numerical coefficients, which won't be important. We agreed there's a constraint for unambiguous decoding, which means that the period at the I plus 1 scale was equal to, by this definition of the grid scale ratio, period at the ith scale divided by the scale ratio, this must be bigger than the size of the firing field. I'm not going to revisit why we said this. Please accept that I said this. This means that lambda I over Li, these two things, must be bigger than the scale ratio for unambiguous decoding. I want to point out the number of grid cells that you have here is proportional to lambda I over Li. If you wish to minimize the number of grid cells, right, you should set lambda I over Li, period, for field width equal to the scale ratio. That's the way you get the smallest possible number of cells. And the reason why you might want that is, of course, a very important resource for any neural circuit is how many actual elements you have, how many circuit elements you have in it. Because every circuit element wants power, wiring, this space, that takes up lots of resources. In fact, there are papers by Atwell and Laughlin from about 20 years ago showing that the dominant cost in the brain is just sending ions back and forth when neurons communicate or compute. What you want to do, if you want to minimize the power, 20% of the power consumption of a human being, is you want to have fewer cells doing the same computation. So it's a very important resource to minimize. You'd like to minimize the number of cells while keeping the resolution fixed, because that's fixed by behavior. Let's look at that. We agreed you have these parameters Ri, which are the ratios of scales of these grids at different, of these periods of these grids at different scales, or the resolution is a product of these numbers. And n, the number of grid cells, is proportional to some of these numbers. Now our problem is defined. I've again left out the constant here. We want to minimize n, the number of cells, so that's the sum over Ri, keeping the product fixed. Easy. And the solution is the following. The first thing is that the ratios of adjacent periods will be equal. Right? That's like a base n number system. What's the base? The base turns out to be the optimal ratio, grid scale scale ratio, is the deep root of E. In D dimensions, you want to have the base, the optimal base for representing numbers is the deep root of E. Now, so this work was done by Shui-Shin Wei, a really terrific student of mine. He was a math student and then moved into cognitive science and then did his PhD with me. He's a professor at UT Austin. I think applied math and neuroscience. So it took like 10 minutes to work out on the blackboard. And then we looked at each other and said, that's really beautiful, but it's never going to be true. There is nothing ever in biology that works out so neatly. Wait and see. So in truth, if you want to compare with data, you have to be a little bit more serious. Really, you should talk about neurons firing in some probabilistic way in some over wider region. So at each scale, what you really know, some probability distribution about where you are. You combine those probability distributions over scales, blah, blah, blah. You can do Bayesian decoding. You have to get a little bit more serious about all of this analysis. Also in 2 dimensions, there's a doubly periodic lattice, and you got to optimize that and stuff like this. And you can do all of that. And here's what you find. The simplistic model that I described predicts a scale ratio between a whole series of grid modules, square root of E for 2-dimensional navigation. Probabilistic theory, because of the little overlaps of the tails distributions, gives a slightly smaller number, as it turns out. That's like 1.45. This was a data set from about 18 years ago. They had like 3 cells. This is a data set that has like hundreds of cells. By now, there are 2 or 3 more data cells within the thousands of cells. And look, that's a beautiful match. It's about as beautiful a match as a theory is ever going to get data in biology. So personally, I would say that evolution seems to have invented baseline number systems and then optimized them for neural hardware. At least that's the way I'd like to say it. And furthermore, it's heartening that it appears that an efficiency principle, which many theorists are fond of, seems to explain the organization of complex circuits supporting a cognitive function. The next thing I'm going to talk about, I think I have 15 minutes. Yeah. Next thing I'm going to talk about, great, we have this map, and it appears to satisfy some of these ideas of somehow representing space efficiently. And actually, these maps are formed dynamically. So here's some evidence for that from the data. So initially, when an animal enters a new room, if you're recording from a cell, there's no grid map. But what actually happens is that if you come to a normal room and you're sort of floating around, you'll get emission. This is the black lines. It's the trajectory of the animal. You'll find as it moves around, the cells fire wherever they fire. And eventually they settle down to this sort of grid pattern of firing. Somehow self-organized or learned during navigation. What's more, it's clear it's anchored to the world, because you can put a cue, like a red card, in the room, and move the cue, and the whole grid will rotate. It's also true that it's not just visual cues or something like sensory cues, because the grid will persist in darkness. You can turn off the lights, remove as many cues as you can, and for about half an hour, the grid will persist in its merry own way. So clearly, you can use self-motion, right? Integrate your path, integrate your motion, maintain this grid. It's also important. We'd like to have a network of neurons organized roughly like in the entorhinal cortex. How can it self-organize a dynamical map of this kind? So you want to think then about neurons as dynamical systems. Now, there are 3 major ways people do that in the field today. The first way is called the leaky integrate and fire model. It goes back to a person named Lepic in 1907, same year special relativity was invented. In this model, imagine that a neuron integrates current coming from its inputs. That integration is a little bit leaky. That if it reaches some particular threshold in its voltage change, it goes boom and fires, and by hand you set it back to 0. You start again. That's actually very useful, a simplistic model. The industrial strength model is due to Hodgkin and Huxley, who won the Nobel Prize for this work in 1963. You haven't read their experimental papers, you have to. These are heroic, absolutely heroic work done before the advent of oscilloscopes, even. A really amazing work. And what they do is they model all the stochastic nonlinear biophysics of all the ion channels and receptors and what have you in the neurons. And they'll give you beautiful spikes that exactly describe the shapes of the responsive neuron. This is like majestic. It's also extraordinarily difficult to work with because there are a bazillion parameters. You never know the parameters for the neurons at hand. You put it on a computer. By the time you put 10 of these together, your laptop will give up. Stuff like this. It's hard to work with. Just in the last 15 years, Eugene Izikevich, who's he's at UCSD, I'm not sure actually, has done what we would call in physics an effective theory of this dynamics. recognize that in the end, all these parameters together implement a dynamical system, various kinds of patterns of bifurcations and attractors and limit cycles and what have you. That's what they're doing. Along the way, they make these spikes. He built an effective model with just 3 equations and 4 parameters that can mimic entire space of behaviors you see from different kinds of neurons if you fiddle the parameters correctly. So in my group, for example, this is what we use when we want to have realistic neurons because you can handle it. And it's good enough, right? You want the effective behavior. So we are going to mostly use a leaky integrate and fire neuron for our purposes. I'm going to tell you how a single grid module is self-organized. And here I'm going to follow some work by Yoram Burak at Jerusalem, Ilafit with MIT, from about 17 years ago. Imagine that this is the entorhinal cortex. The blue dots are all neurons, excitatory neurons in this structure. Now, further imagine that there is some external drive that comes in and causes each of these neurons to fire. They're all separate from each other. What's going to happen? They're all going to get driven. They're going to go brrr, some firing rate that depends upon their driving. Next, imagine that each neuron inhibits its neighbors. There's lateral connections where each neuron tries to shut its neighbors up. That's faithful to the actual circuitry you see in this part of the brain. You have lateral inhibition. Now you can see what will happen is that if this neuron fires, it's going to shut its neighbors up. The next neighbor beyond that is not shut up, so they'll fire. It'll keep going like this, and you'll get a pattern of firing. You can work out the fixed point of this dynamics as a triangular lattice. Not surprisingly, it close packs the structure of firing. However, when you produce this triangular lattice, you're going to have a continuous manifold of phase-shifted attractor states. Why? You broke the symmetry, translation invariance, and there's going to be the zero energy mode that shifts things around. It's going to be unstable to slipping in this way. So the idea of Burak and Feet is to couple, actually there were people before that, Tourettes and Fuse are also, should be given credit for this. The idea was to couple these zero modes to the motion of the animal. And what you do, so you have the head direction of the animal and the speed of the animal. I told you there are cells that encode head direction and speed. You feed that into the circuit, into the differential equation controlling the circuit in the correct way. And then what you'll find is once you fed that in, if the bright dots here are the pattern of firing on the neural sheet, you'll find that this pattern slips and moves around as the animal moves around. So the consequence of that is if you look at this red dot is here supposed to represent one neuron on this neural sheet. As this pattern of activity shifts on the neural sheet, sometimes it's over the red dot, and sometimes it isn't. When it's over the red dot fires. So in effect, what's going to happen is that this neuron will fire when the animal itself is physically on a triangular lattice of locations in the world. Basically, the coupling with the motion of the animal basically produces, if you like, the isomorphism between the neural sheet and the world, right? And there you go. Right? So that's what you wanted. Now you have a map of the head. Your head is holding where you are in the world. Now, the problem we have, that was old work. The problem we have is we have to make a series of modules. That was done in the early days when people didn't even know that there was more than one module. How do you make multiple modules? And what's more, you need discrete changes. Right. So one idea might be, I take this series of sheets, and then I increase the scale of the inhibition beat scale. So, so basically you shut more people off around you. If you do that, you'll make the grid pattern bigger and bigger and bigger. The challenge is that if you do that, you're going to get a continuous, if you do that continuously, you'll get a continuous change in the scale of these grids. And what you want is a hierarchy of discrete modules. So one way of doing that could be developmentally. What you do is you just wire it so that you have this scale here, and this scale here, and this scale here. Seems a bit clunky, but you could. We want an alternative idea. So there's an alternative idea that maybe you could self-organize it. The analogy, so this was work done by another student who was a condensed matter theorist. And the idea was an analogy with something called the Frankel-Kontarova model you can find in the book by Chaikin and Lubensky. Seems to be missing from our library. So the Frankel Kontarova model works like this. There's a substrate which produces a potential for some period. Then you have some other particle that's going to stick to the substrate. There are interparticle interactions. Now, the substrate wants to stick to the particle, and the interparticle interactions, let's say, want to stick in a different way at a different period. So there's going to be competition, a push and a pull. And depending upon the balance between this push and the pull, you can wind up getting very different organizations in how the whole absorbed lattice works. Okay? Why don't we try that? We have already each scale a push is pushing the pattern of firing apart. If we now ran wires between each of these layers, so that each layer tries to maintain the same periodicity at the next scale, that's like these potentials, trying to make the absorbed things having exactly the same period, then you would have a push and a pull. In fact, an unknown thing in the study of the enterinal cortex was that people had noticed that there are wires going along the structure. People didn't know what it was for. We can invoke it. If you do that, do some work, you'll find that you get these discrete modules. Kurt is telling me I have 5 minutes left. So I'm going to probably skip. No, let me. So you find you have discrete modules. The parameters that control these discrete modules not to be how much push and pull you have. How strongly you try to force the next layer, next module to have the same period versus how much you try to push them apart. And you can work out that there's a phase structure here. Depending upon the relative strength of these different things, you can get any different phases in which these different lattices organize in different way, just like you can get in the Frankel Kontarova model. So here's the experimental evidence that this is a reasonable idea. First of all, it turns out that this model predicts that the grid orientations are discrete. You don't get all possible orientations. That's true in the data. It also predicts that the relative orientation of modules will be preserved in individuals across different orientations, because the relative orientation is determined by the coordination between these layers. It's also true in the data. What's not true is that the precise angles that are predicted are not precisely the same as in experiment. But we have actually left an important thing out. All of these systems, in fact, also interact the boundaries of the space. I mentioned that, right? If you have a queue, you can move it around. And I also mentioned there are cells that encode locations on the boundaries. If there's any interaction with them, that will change what you predict in the absolute orientation. That's been left out here. So I have, 3 minutes? Let's see. How much of this can I do? I can do 3 minutes worth. So in fact, there's evidence also of these interactions at the boundary. For example, I've just touted elaborate model of self-organization of these grids. What I didn't tell you is if you just run it forward, it accumulates errors. There's positional drift because real neurons have noise in them. What you can do is you can imagine you also have border cells, and that there's some sort of learning that goes on, Hebbian learning, where neurons, border cells, are also present in these structures. The fire an animal at some location near the boundary, suppose you imagine that they increase their synaptic strengths with grid cells that fire at the same phase. You could ask, well, that could be an error correction mechanism. It's like a bumper mechanism. The grid is drifting, and you arrive at a particular point. What do you know? You have a bumper mechanism that corrects any drifted phase. So you can show that indeed that fixes things and reduces the drift. That theory then makes a prediction. I'll tell you the prediction, show you the data, and that's the end. Whoops. Prediction is the following. If it's really true that these grid systems are anchoring the boundary in this way, the obvious thing to do is to change the space, stretch it, shrink it, like, in that Star Wars scene where inside this room shrinking. Because if you do that, you should untether the phase of grids with boundaries. And that should produce an effect depending upon whether the last wall you touch, the right wall or the left wall, the whole pattern of the map shift left or right because it's tethered that way. You can build a model of that. This is the data from these labs. which shows precisely such a shift. It's hard to see a little bit because real data is always noisy. You have to do this with statistical analysis. So Gordon is doing this, which means he doesn't believe the data yet. We'll look at this, which is supposed to show the relative shift in the after left wall touch and right wall touch. You can ask me in more detail afterwards, or at the, in a minute, and I can go through that in greater detail. You have to do some careful statistical analysis in order to be able to establish that it's a statistically significant effect, because the responses of these cells, this is just one cell, are pretty noisy. So it's hard to make, so you have to do some work to make sure it's a statistically valid effect. Claim here that the predicted effect is seen. This is the end. Let me end with the following things. There's lots of more things to do. So, for example, I talked about self-organizing grid responses. But I also told you there are border cells and head direction cells, etc. I didn't tell you how those self-organize. That's a thing to do. Another thing is almost all experiments are done in little squares and circles and things. Anybody who's seen a real burrow of a rat or something knows it's very complex. Well, look at this room. So actually, I have a project on with Dori Dirdickman, a colleague, a friend, and Shai Abramson, a student at the Technion, where we now have Shai constructed a rig where you can modify the environment at will to complicated shapes, like, you know, with burrows and tunnels and things like this. And you can change it on the fly if you realize that you should modify it with the animal. And so we'll have results in this soon. I think I completely left out is that these maps in your head reload in a context-dependent way that are complete. How does that even happen? I have a student, Spencer Rook, also formerly at University of Illinois, Urbana-Champaign. He's a math student originally who's working on that. Then there's things like optimal navigation. The purpose of all of this is to navigate. If you navigate optimally, do you develop these structures? It's a machine learning question, right? You can get a robot to navigate, ask with the correct constraints. If you ask for it to navigate, do the artificial neural networks develop representations like this, because it's efficient. So I have two students working on that. And the final thing that I'm actually really interested in, also working with Dori's lab, the egocentric experience versus allocentric maps. Let me explain that. So your experience of the world is relative to self. It's egocentric. But the maps that are described are allocentric, relative to some absolute landmark. How does that transformation happen? Especially because to act in the world, you translate allocentric map to egocentric action again. That's a really interesting question, especially because there are languages. For example, these are 3 languages of Australia. People never use egocentric language, by the way. You wouldn't say the ice cream store is to the left. You would say it's to the north. You would even do that inside closed rooms. We maintain that. So we clearly have access to both these kinds of information. Very interesting to piece that apart. Anyway, I hope I've convinced you this is an exciting and fruitful source of problems for us.
So we have time for some questions. Thank you very much, Vijay, for disorienting talk.
That's it.
No, now I'm thinking about it. Why am I where? How do I know where I am?
It's a good question.
So, you recognize the question?
Yeah.
Yeah. Repeat the question for the.
Julio.
Thanks for a super interesting talk. Do birds have a 3-dimensional version of this? And did you see 1 over EQ to 1/3 or something?
So Julio Paramartinez asked an excellent question. Just do birds find 3 dimensions, have a 3-dimensional version of this, and what about the cube root of E? Now that's a good question. And there's a similar, there's also, you can ask this question, bats do that. Data on these grid cells in birds is not available yet, because you got to mount the thing and it has to stay there while the bird flies, all challenging. They've done it for bats, and they don't quite have enough data about the modules and how many modules there are, etc. So it's not completely clear. Also, as it turns out, bats actually tend to spend a lot of their time crawling on the surface of the wall of the cave. It's really 2-dimensional. Something intermediate is happening. The grids look as though they have some intermediate quality to be explained. Good.
have the same question, but I have a follow-up too. Have you done theoretically the generalization to where, let's say you're in 3 dimensions, but you need to map one of the 3 differently? So for example, you might think birds want to perceive X and Y very differently than Z, the height. Is that a theoretical work that's done?
No one has done that. It is an Razio asks, if you're a bird or a fish, for example, or a bat, the X, Y direction may be very different in what you want to do with it, and the Z direction, because in X and Y you're navigating from place to place on the Earth, and Z direction is just how high you are, and it's just empty open space. You don't care to map it that carefully, for example. No one's actually done that. I think that's a completely pertinent question. The reason I'm sure it's a completely pertinent question is there are weird reports about the spatial abilities of things like squid, deep sea squid, the stuff that lives in the deep ocean where there are no borders. I had a colleague at Penn who was explaining to me that she worked on squid eyes. It was impossible to keep these things alive in the lab because they would just go full speed at a wall and bash their head. They would do it again and again and again until they died. Right? They just didn't understand that the space had a border. So, so you're saying points to the fact that depending upon the behavioral niche you're in, you may have different requirements with this kind of thing, and that has not been studied by anybody. Ramita.
Good question. So the original tiling that you showed, so you go from, you split the square or the line in 2, and then you go half. So somehow assumes that the space is somewhat homogeneous, so you can't put everywhere the same way. If it's not, Shannon has told how to deal with optimal encoding. Is that something?
Super interesting. So Ramita Nur Muhammad asks, what happens if the space is not homogeneous? So for example, you could do it through curvature. You could have a little bump here, like in the real world, things like that. Or you could make it inhomogeneous through having targets in different places. There's the food there, and there's the, I don't know, something horrible there, shock or something. I don't know. So what happens? So you should separate that into 2 parts. So there's the entorhinal cortex and the grid cells that I was talking about. Then there's the hippocampus with the place cells that I mentioned, and we should mention both here. So with the entorhinal cortex, what does happen is that if you deform the space, the grid fields do deform in different ways. So they respond in part to your self-motion. So if I'm looking from above and this is the square, over here, it's actually more space because you put a bump there, the grids deform around that. So I think there's one paper on that, Alessandro Treves from TISA, but it really hasn't been studied. Experimentally, it hasn't been studied as much, but this would be easy to do. And so that's definitely in people's minds. There's just so many more pressing questions, but it's an excellent question. Hopefully resolve. One thing that is true is that if you squish the walls of space in some way, it's known that the grids deform in some particular manner. That seems to be somewhat consistent with this theory of anchoring to the boundaries that I was talking.
One more question from this side of the house.
Yeah, okay. And I'll take one for that. Yeah.
So if the space has a curvature, is there a cell type that attends to that curvature?
Yeah, that's a good one. So there are recent reports.
Repeat the question.
Oh, yeah. The question was, are there cell types that attend to the curvature of space? And there are very recent reports as of this year that there are cell types, claimed cell types, that attend to the curvature of boundaries, not of the bulk space, because that just hasn't been studied, but of the boundary of space. So that's something that, everybody's digging into, why, wherefore, etc. Yeah.
Your coordinates are the room coordinates.
Yes.
But you showed a model of self-organization, and those coordinates seem to be the neuron coordinates.
Okay. So what I was trying to so the question was, the coordinates of the map are room coordinates, but then in the self-organization description it was neural coordinates on the sheet. What's the relationship between those? So sounds like I should have explained a little piece of what I said a little bit more, and it's like this. So there's the neural coordinates, there's the world coordinates. What you need is to produce a map between them. You need to connect them. The way this is done is in that circuit, there are also cells that different cells that respond to different head directions, first of all. So this way, this way, this way. They sort of piece out the directions your head is pointing in relative to some absolute landmark, like, I don't know, a point on the wall. There's also cells that have different firing rates depending upon how fast you're moving. This is some knowledge you get from knowing something about your own motion. You can feed these two pieces of data in, take the neural map, or the map of firing on the neural sheet, and translate it as you physically move. The data about your head direction and your velocity is used to move the pattern of firing, literally move the pattern of firing on the neural sheet. And that's what produces the association in neural coordinates and space coordinates.
All these coordinates are correlated. Those neurons are geometrically.
Yes. Yes, though it doesn't actually completely follow that it's the so I've sort of straightened out the sheet. I've put things next to each other in a particular way. Really, there's some defects in the sheet, and it's sort of organized in a somewhat funky way. But right, the two-dimensional sheet, roughly, there's a two-dimensional sheet. You need to make them the same two-dimensional sheet. That's connected through the data about your motion connects them. You can take some absolute anchor here, and you just need to move them around. Isn't it cool? You had a question. Then Devaki. Yeah.
No, I was wondering that this is fantastic. Have there been any studies for other brain functions like smell?
Yes.
Do they follow this grid pattern?
They don't, as far as we know. So, the question was, do other neural functions like smell, sound, vision, etc., fall to this function? There may be grids in some place. Let me tell you just a snippet about each of those. So, vision, what happens is that I described the retina. The end of the retina is just like a computer vision program. The output of the retina is a collection of features extracted from the world: local motion, color, bright spots, dark spots, stuff of this kind. I have another talk telling you all kind of really cool stuff about the adaptation of those things to the structure of the visual world. It's a really astonishing story. Anyhow, you get these things, send them to the brain. Actually, you send them to a part of the midbrain called the lateral geniculate nucleus, where the searchlight of attention gates what goes through, blah, 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 blah. At the end of the day, the signal arrives in the visual cortex. The visual cortex, the process of reassembly begins. So in the first layer of visual cortex, edge detectors of different kinds. Just like if you open up a GIMP or one of these computer vision programs, you'll find Gabor filters pulling out edges. Literally Gabor filters, simple cells. Gabor filters were first invented for describing edges in the world because it turned out they fit simple cells responses in the visual cortex well. So Kuhl and Wiesel won the Nobel Prize for discovering these things. Then in V2, these things are assembled. You do textures, color. Then these are assembled in V3 and V4 into corners and things like that. Then the signal bifurcates. It goes up to the motion pathway. For our purposes here, the shape pathway is sort of goes down this way. There's an area called the inferior temporal cortex, where you'll have cells that respond to particular shapes, no matter where they are in the visual field. For humans, there's a little sub area of that, the fusiform face area, that does faces. The reason I'm saying all this is that 6 synapses from the eye. And if you look at any deep network, you'll have this enormous network with bazillions, millions of data stuck into it for it to learn faces. We can recognize faces after one viewing, 6 synapses. There's something very cool about this. It's vision. Vision is very different from olfaction. The reason it's different from olfaction is really a method of feeling the shapes of molecules, right? The plants and animals put out various kinds of smells. They're combinations of volatile molecules. And you need to design a system that can sense these. We'll have another talk on that. And so the way this works is in your olfactory epithelium, there's a certain repertoire of receptors. For humans, it's about 300. Mice, it's about 100. Elephant is about 2,000. The fly has about 50 to 100. We're all a few hundred, give or take a factor of a few. It's interesting as a fact. So then what happens is that every molecule binds to many receptors, and every receptor binds to many molecules. You get this heavily multiplex signal. And this multiplex signal seems to get further randomized, actually, in the projections to the brain. It's literally a random pattern projection. So there's a whole talk that I give on the theme of the olfactory system being randomness, because if you randomize signals like this, you can get very compact distributed representations of that data. It's a different kind of data in the world. I think this is an adaptation to the kind of data that's coming in. Audition is still, again, different because, basically the ear basically does a Fourier transform, right? So it splits up signal to frequencies, wavelets actually, and splits them up, sends them to the brain. And the brain, you have a structure where the different frequencies are laid out, kind of, just about. And then from there, the kinds of problems that happen olfaction and audition are like the cocktail party problem. Many people are talking at the same time. We can pick out, your voice or your voice, something like that, clearly after one hearing. How does that happen? How do you learn in that sense what olfactory and auditory objects are when they don't sort of occlude each other like they do in vision? So that's another class of problems. And the question you're really asking is, why is this kind of grid representation efficient for this and not for that? And we could go into questions of how quickly you need the information, what to do with the information, dimensionality of the information. This is three-dimensional information, right? Or two-dimensional information. Those things are extremely high dimensional. The olfactory space is like 10,000 dimensional. So it's much, much higher dimensions. That changes the nature of the problem. Devaki had a question.
For babies to adults, so you start with egocentric and move to autocentric.
Okay, so Devaki Bhaya has a very interesting question. You have these structures already as a baby, and as a baby, maybe it's just egocentric, and then as you grow up, it becomes allocentric. Very interesting question that is not resolved. So the data that's available is almost all from mice, a little bit from rats, right? And the data that we have, we know essentially zero about the relative proportion of egocentric, allocentric, what have you, in the infant, right? What we do know is in the infant mouse, the grid cells don't appear to be formed, right? They don't seem to make any grids. They seem to have some kind of place cells. And those place fields tend to congregate a little bit near the boundaries of the space or near landmarks like their mother, right? So that has led to debate in the field involving, do place cells make grid cells? Is the source of the signal coming in to drive the grid cells to the place cells, or do grid cells make place cells? And there's a very interesting set of work being done, including my own group, on imagining that the place fields involve, basically anchoring to the boundaries initially. They involve memory and reloading your map, that they then trigger something in the grid fields. And then the grid fields, once they develop in the adult, go back and repair, fix, and improve the place map. That's the current thinking. There's a combined experimental theoretical problem we said there. I just realized that I didn't answer part of Armita's question. Yeah, and there's a part of your question which I didn't answer, which involved place fields. And it turns out that there are place cells that I didn't get a chance to talk about today. Those are definitely inhomogeneous in space. So for example, there's now ample evidence that if you've got goal locations, they congregate there, right? Important places are better mapped inside the head. That sounds like a perfect, efficient coding problem. I have a repertoire of n place cells. How should you invest them in terms of having resolution in space? It sounds like it's a problem made for my group. We should work on this.
Okay, well, fortunately, we have cookies and other refreshments available, and we can send on Vijay and ask him more and more of these questions. So let's thank him now for.
Thank you.